Welcome to this explainer. Today, we're diving straight into the incredible architecture of life itself, proteins. We're going to build them from the ground up, starting from their absolute smallest biological Lego blocks, all the way to their final, highly complex and functional forms.
Here is our biological roadmap for today. First, we'll meet the raw materials, the amino acids. Then, we'll see how they link up via peptide bonds, watch them fold through 4 distinct levels, and finally, uncover the invisible chemical bonds that hold the whole thing together. Let's get right into it.
Okay, let's set our baseline. Proteins are essentially biological polymers formed from the condensation of individual amino acid monomers. Think of amino acids as the 20 individual letters of an alphabet. By linking them together, you can form near infinite combinations of complex sentences, some literally thousands of letters long. In fact, there are tens of thousands of different proteins just in the human body alone. Without these incredibly complex structural and functional molecules, life as we know it simply wouldn't exist, because they drive everything from cellular repair to digestion.
Let's start with step 1, meet the amino acids. To really grasp the whole protein, we first have to zoom way in and meet the basic building blocks that make up that biological alphabet.
Every amino acid shares a common core attached to a central alpha carbon. You've got an amino group, which has basic characteristics, an acidic carboxylic group, and a simple hydrogen atom. But they differ completely based on one crucial element, that unique R group side chain hanging off the core. That variable R group is the ultimate wildcard. It's exactly what gives each of the 20 different amino acids its own unique chemical personality.
Now, you might be wondering, how do these specific chemical groups behave when they're actually inside a living cell? What happens to these molecules when they're swimming around at a standard cellular pH of roughly 7.4?
Well, the answer is highly dynamic. At a cellular pH of about 7.4, amino acids exist as zwitterions. That's just a fancy way of saying they become bipolar ionic molecules carrying both a positive and a negative charge at the exact same time. The amino group actually picks up an extra hydrogen to become positively charged, while the carboxyl group loses a hydrogen to become negatively charged. It's this dual nature that allows them to interact so seamlessly in the body's watery environments.
Moving right along to section 2, linking up. Now that we have our raw materials, let's head down the assembly line and see how these individual monomers actually connect to start building a chain.
The absolute crucial mechanism here is a process called condensation. When the carboxyl group of one amino acid and the amino group of another bump into each other, they interact by dropping an oxygen and 2 hydrogens, releasing them as a single water molecule. This exact removal of water is the catalyst that allows the 2 amino acids to chemically lock together.
And boom, this process builds a sturdy, new connection known as a peptide bond, officially turning our separate monomers into a dipeptide. You get this newly formed carbon-nitrogen bond acting as a bridge to hold them tightly together. This exact repeating mechanism is what builds massive polypeptide chains out of hundreds or even thousands of amino acids.
But biological systems also need a demolition crew, right? If a cell needs to break down a protein to recycle its parts, it performs hydrolysis. By literally forcing a water molecule back in, it severs that exact same peptide bond. Adding water breaks the link, returning the dipeptide back into 2 free-floating amino acids.
Let's jump into section 3, folding. With our peptide bonds forming a long polypeptide chain, it's time for the packaging and shaping phase. A simple straight chain of amino acids isn't a functional protein yet. It has to fold perfectly to actually do its job.
It is fascinating to trace this entire architectural journey. It starts from a simple primary string of amino acids. Think of this as your long, unbroken sentence. Through hydrogen bonding, that primary sequence twists into secondary structures, either forming folded beta-pleated sheets or spiraling alpha helices. Next, the whole thing collapses upon itself into a highly complex 3D tertiary shape. And finally, some proteins take it a step further to the quaternary level. When multiple folded polypeptide subunits join forces, they achieve that ultimate quaternary structure. Take collagen, for example. 3 distinct chains twist together like a mighty rope to provide incredible structural strength for your skin and bones. Then there's hemoglobin, which combines 4 distinct chains to form a complex molecular carrier perfectly designed to transport life-giving oxygen through your bloodstream.
Finally, section 4, the bonds that bind.
We've seen these beautiful and intricate 3D shapes, but what invisible forces are actually holding these folded structures together so they don't just unravel? The 3D structure is stabilized by a specialized team of interactions. We've got weak forces like hydrogen and ionic bonds acting between charged R groups, working right alongside strong covalent anchors called disulfide bridges, formed specifically between cysteine amino acids. We also have hydrophobic and van der Waals interactions, where nonpolar R groups huddle together in the absolute core of the protein, hiding away from the watery cellular environment outside. All those specific R groups we met earlier, they're chemically, almost magnetically attracted to each other, forcing the protein backbone to buckle and fold into place. You get hydrogen bonds bridging gaps, ionic bonds pairing up opposite charges, and solid disulfide bridges acting as chemical staples. But here's the catch. These structures are incredibly fragile. If the surrounding temperature gets too hot or the cellular pH drops or spikes too extremely, these delicate bonds snap. This causes the protein to completely lose its shape in a process called denaturation, permanently destroying its biological function. It is literally like melting a key so it no longer fits its lock.
Ultimately, all of this precise chemical folding results in two main classifications: long structural fibrous proteins like collagen, or tightly coiled, highly functional globular proteins like hemoglobin and your body's enzymes. Our entire biology relies on this flawless architectural sequence. But knowing just how fragile these folded structures are, it leaves us with a pretty profound question to wrap up on. How does slight, tiny errors in this primary sequence, just one single wrong letter in the alphabet, cause massive, cascading biological diseases down the line? Something to think about until next time.